Hello, good morning everyone. Today we are going to understand Lac Operon concept. Lac stands for lactose sugar. Lactose is a disaccharide. It can be broken to form single units which are called monosaccharide. Units of lactose are called glucose and galactose. Operon refers to set of genes present in the bacteria which will be controlling one function like lactose metabolism. How it differs from eukaryotes? In eukaryotes, for one function or to produce one protein, there should be one gene, one protein. But when it comes to prokaryotes like E. coli, many genes together they function to control uh, one metabolism like lactose metabolism. So that concept is Lac Operon. The Lac Operon concept was given by Jacob and Monod in 1960s. Let us understand. So through Lac Operon we are going to understand when there is no food in the medium, how genes are switched off. Because if there is no food, unnecessary production of enzymes are not at all required, so they should switch off their gene. How it occurs? When there is food in the medium, food is there, so it has to be metabolized. For that they have to secrete enzyme. How they switch on their genes to produce those enzymes? That is the overall regulation of gene in Lac Operon concept. First of all, if you take the bacteria contains DNA. I have enlarged a segment of DNA. Segment of DNA is called gene. It is an operon where many genes are going to control one particular function. What are the genes present in the operon? There is a promoter. Promoter is a site where RNA polymerase enzyme comes and binds. RNA polymerase has the capacity to it will stimulate the gene to undergo transcription to produce mRNA. So there are two promoter, one for I gene. I refers to inhibitor because it is going to produce an inhibitor protein. One more promoter is there. It is to regulate operator gene. Operator gene controls all other structural genes. There are three structural genes: LAC-Z, LAC-Y, LAC-A, whose role is to produce enzymes required for lactose metabolism. Hence, operon is composed of promoter and there is inhibitor which is going to produce repressor protein. One more promoter which will regulate operator, as well as there are three structural genes: LAC-Z, LAC-Y, LAC-A. That is about the structure. Now let us observe when there is no lactose in the medium. Lactose is called inducer because presence or absence of lactose will induce, or it is the one which is responsible for switching on switching off of genes. So when there is no inducer in the medium, promoter comes and binds RNA polymerase comes and binds to promoter. Now it will transcribe the I gene. I refers to inhibitor. So there is a production of mRNA. mRNA undergoes translation to produce a protein which is called repressor protein. Repressor protein, as soon as it is formed, it has got a binding site on the operator gene. It goes and binds to the operator. Now operator, when it is attached to the inhibitor protein or a repressor protein, RNA polymerase tries to transcribe the structural gene, but it cannot move further. After operator point, it cannot go further. Why? Because operator gene has been blocked by the repressor. When RNA polymerase cannot go towards the structural genes won't be able to produce anything. In this condition, genes are said to be switched off in operon. Now let's consider that to the medium we are adding lactose, or we are providing the food for the bacteria. So I have written in the presence of inducer. Inducer is lactose. lactose is there outside, and in this plate bacteria is growing. This is a bacteria, its genes are shown. What happens? When there is lactose in the medium, few molecules of lactose will enter into the cell of bacteria. How does it enter? Because there will be presence of little amount of permease enzyme. Already it will be there. It doesn't mean that 100% permease enzyme will be there, or little bit will be there. Using that, lactose molecule will enter into the cell, and once it is inside the cell, lactose is called allolactose. Allolactose, what it does is, repressor which is attached to the operator, now allolactose will pull the repressor protein by attaching to it. Okay, what did I say? When the lactose enters into the medium, it will, by attaching to the repressor protein, it will pull repressor protein out. So now operator is free. RNA polymerase attached to the promoter, as well as RNA polymerase can move further because operator is free. There will be, so they start transcribing structural gene. Genes in prokaryotes are said to be polycystronic gene. What is polycystronic gene? Many genes together, they will be producing only one mRNA. Whereas in eukaryotes, if there was 3 genes, they would have produced 3 mRNAs. So many structural genes are there which is going to produce one mRNA, but one mRNA can produce 3 proteins. That is the meaning of polycistronic. Many genes producing one mRNA coding for many proteins. So they will code for 3 proteins which are known as beta-galactosidase, permease enzyme and trans-acetylase enzyme. So enzymes are nothing but proteins. Beta-galactosidase enzyme, whatever lactose which has entered, it will break the lactose into its monosaccharide units, glucose and galactose. Glucose can be utilized. Further, more and more lactose will enter into the cell with the help of permease. Name permease means it makes the cell permeable to the entry of lactose. As more the lactose enter, more the galactosidase is secreted, it will be broken to its monosaccharide units. That's how food is taken by the E. coli bacteria. Trans-acetylase enzyme. Trans-acetylase refers to, it will help in transfer of acetyl group during the process, which will enhance the effect of beta-galactosidase and permease action. So in this case, when food is there, genes are producing their enzymes, metabolism is going on. Since their genes are functioning, we can say genes are said to be switched on. When it is switched on? In the presence of inducer. When there is no inducer, genes are switched off. Again, if the lactose gets over, then repressor binds to operator, again it will be switched off. So this is how gene regulation in E. coli occurs in the presence or absence of lactose. Now what kind of regulation it is? When there is repressor protein production, genes are inhibited. So in the presence of repressor protein, it is said to be negative regulation. In the presence of lactose, genes will be expressing. So presence of lactose indicates positive regulation. Lac Operon is under negative control regulation, as well as it shows positive regulation depending upon the conditions. So this is how Lac Operon operates. I hope you have understood the concept. Thank you for watching.